Hi to all of you. Welcome to this video. We'll be discussing nucleic acid chemistry in this lecture, which comprises of the structure and composition of the two different nucleic acids, DNA and RNA. Then we'll be discussing Watson-Crick's model of DNA and Chargaff's rules. In the first lecture, we have covered that both DNA and the RNA are actually the polynucleotides, and depending upon the sugar moiety, we categorize them as such as deoxyribonucleic acids, which contain the sugar as deoxyribose, and RNA contains sugar, which is ribose only. Out of these two polynucleotides, DNA is most important because it makes up the genetic material in all the living cells and tissues. It contains the information which is stored in it and then which is replicated, transcribed, as well as then translated for the cell so as to function and exist. So DNA component usually is a part of chromosome or chromatin. We know that, and the second part of that chromatin or chromosome is the protein. We have covered it in the molecular biology already. You need to see that lecture also in order to be sure about the chromatin structure. So by 1950, it was known to the scientific world that DNA is a polymer of deoxyribonucleotides, and it is linked through phosphodiester bonds. So the essentials of the DNA were known. It was known that the monomer unit which is repeated in this polymer are mononucleotides, which are of deoxy type, and the backbone is made up of phosphate and sugar. And because of this phosphate and sugar and the asymmetry of the mononucleotides, the two different strands of the DNA was having a different and distinct polarity. So this is the structure which is given for the DNA, which is based upon now called as the Watson and Crick's model of DNA. So the current version of the DNA, which is a double helical model, was given by James Watson and Francis Crick in 1953, and they gave their model based on the known structures of nucleotides. However, the data which was used for this model was actually the data of Rosalind Franklin and Maurice Wilkins, which was the X-ray diffraction data of the DNA fibers, as well as the chemical equivalencies given by Chargaff. Actually, this was the classical case of the scientific theft which Watson and Crick pulled through and utilized the data from the Rosalind Franklin's diaries to come up with this model. Both Rosalind Franklin's diffraction data as well as the model for the DNA was published in the same issue of the Nature. However, we usually tend to give credit to Watson and Crick only, but actually the credit goes to the Maurice Wilkins and Rosalind Franklin for discerning the actually spatial arrangements of the nucleotides in the DNA. So what are the salient features of the Watson-Crick's model of DNA? First and foremost, DNA consists of two strands. They are wound around each other in a double helical manner, as you can see in the accompanying picture. Usually it is right hand helix, and the two strands are antipolar to each other. That means that one goes in a direction of 3' to 5', and another has the 5' to 3' direction. As you can see also in the molecular biology lectures, the antipolarity is important in the metabolism of the DNA, especially in the replication as well as transcription. So the antipolarity is important. Then the whole diameter of this duplex is around 2 nanometers, and in these 2 nanometers one complete helix turn is about 3.4 nanometers, and in this complete one turn there are around 10 bases of nucleotides. So this is one form of DNA which is the most common, having 10 base pairs of the nucleotides in one complete turn. As you can see in here, this is the one complete turn, and in this one complete turn around 10 base pairs are located and stacked upon each other. So the difference and the distance between the each pair is about 0.34 nanometers in the DNA, this form of common form of DNA. And as we already told you that deoxyribose and the phosphates form the backbone, the vertebra of this DNA duplex. So they are lying on the exterior of the molecule, as you can see also, and the nucleotides are located inside. So to access the information of the DNA which is contained within the DNA, the genetic information, we have to prise apart, we have to take apart or melt the two strands of DNA from each other. Furthermore, the two polynucleotide chains are complementary to each other. That means that wherever adenine is present in one strand, in the opposite strand it will be thymine, and wherever there is guanine in one strand, in the opposite strand there will be cytosine. So cytosine pairs with guanine and adenine pairs with thymine within this duplex structure. Furthermore, the two interactions between the adenine and thymine and the guanine and cytosine are actually the hydrogen bond interactions, and these hydrogen bonds are the ones which hold the two strands of the duplex to each other. Also, the adenine will bind to the thymine via two hydrogen bonds only, whereas GC interact with each other by three hydrogen bonds. So GC are linked to each other more avidly than the AT, and hence during the metabolism, that is DNA replication or transcription, AT sites are the ones which are melted first, and the genetic information resides only in one of the strands. So this strand is called as template strand or antisense strand. This is the strand which is red, and the RNA is synthesized on this strand during the transcription process. And another strand, which I have already told in the molecular biology lectures also, is called as the sense strand, because the sequence of the mRNA which is formed during the transcription is almost similar to this strand, except that there in mRNA there will be uracil and here it is thymine. So the important strand of the two is the strand which is red, which is called as the template strand. Another aspect of the Watson-Crick's model is that when you see the overall topography of the DNA molecule, is its ability to make certain notches when it winds as a duplex. These notches are called as the major and minor grooves. Major and minor grooves are opposite to each other in the topography. Wherever there is a major groove on one side, then there will be a minor groove on other side, and similarly for the minor groove and the major groove. What So what are these major and minor grooves? They are actually opposite to each other in the duplex structure, and they continuously run along the entire length of the DNA molecule, and they are actually the result of the anti-parallelity of the two strands in this duplex molecule. As you can see in the picture, there is a minor groove on one side of the duplex structure of the helix, and then here the major groove is exactly opposite to the minor groove on the other side. So they are actual structure features of the molecule when the molecule is made from the replication process, and they are necessary for the attachment of the DNA binding proteins during the metabolism of the DNA, that is during the replication and transcription, for the binding of certain DNA binding proteins. Further, the hydrogen bonds between the nitrogen bases, that is adenine and thymine and guanine and cytosine, have different kinds of lengths. In adenine and thymine, the bonds are from 2.8 angstroms to 3 angstroms, and the hydrogen bonds between the guanine and cytosine are 2.93 and 2.9 at length from each other. So the interactions usually vary from the 2.8 to a maximum of 3 angstroms. Further discussing the difference between the two different types of nucleic acids, that is DNA and RNA, by that this time you should be able to tell what the difference is. The differences are actually in the four different aspects of it. The first and foremost is the sugar moiety, which you already know. In DNA, it is deoxy type, whereas in RNA, it is ribose type. Stability, in terms of stability, DNA is more stable because of its duplex nature, whereas the RNA, because of its single strand nature, it is less stable. And hence, because of this, DNA makes the most of the genetic material as the repository, as the storehouse of the genetic information, which is then coded to RNA. However, still RNA is in certain viruses make up the genetic material in itself. Then there is a difference between the nitrogen base among all four different kinds of nitrogen bases. The difference lies in thymine and uracil. In DNA, thymine is present, while as in the RNA, uracil is present as the base. Uracil is not found in the DNA, and thymine is not found in the RNA at most of the time. And then the linearity and complexity, which I already told you. In linearity and complexity, DNA is complex and duplex, while as the RNA is less stable and it is simple in structure because of the single-stranded conformation. The Watson and Crick model of the DNA is actually only for one type of a DNA which exists in nature. There are many different forms of the DNA in nature. The three main forms are A DNA, B DNA, and Z DNA. The Watson and Crick model defines the B type of a DNA. What are the other types of DNA and what are their features are given in the next picture. As you can see in the comparison diagram, the B, the central one, is the one which is for the Watson and Crick's types of an DNA. It's called as B form of a DNA, and its helical rotation is right, and the diameter is around 2 nanometers, and the helix pitch, that is one complete turn, makes up 3.4 nanometers, and in one complete turn there is 10 bases. And then the differences in major groove and minor groove. Major groove is here wide and deep, while as the minor groove is narrow, and so on and so forth. While as you will see in the A form of DNA, there is a difference in the helix pitch and the number of bases. Same for the Z kind of a DNA. So A form of a DNA, the diameter, it is much thicker, whereas the Z form of a DNA is packed compactly, has a low diameter among all the forms of DNA. It also contains, Z form of DNA also contains more bases per helix than the B and A form, and rest of the differences are given in the table, which you can memorize. Another salient feature about the DNA is the ability to follow certain rules which were given by Erwin Chargaff in 1950. They are actually the set of conclusions when he studied certain DNA molecules from different sources, and he postulated five different types of rules which are followed by the DNA helix. The three important or the general ones, that is the DNA obtained from the different species has different base composition, that is having a percentage of A, T, G and C, has direct link with the species from which the DNA is obtained. So humans will have a same base composition. Then DNA samples obtained from the same organ, tissue, or of the same species will have the same base composition, that is the percentage of ATGC will have the same composition, whether it is obtained from the human's eye or human's leg or some other cell. Third thing is that base composition does not change with the nutrition, environment, or sex or an individual species. Within this species, if you are getting the sample of a DNA, and let's for example say from the male and female humans, males will have the same base composition as compared to their female counterparts. So, as well as it doesn't depend upon the nutrition. The thin lean will have the same base composition as the obese one. Then the fourth postulate is the most common, which is utilized and given for the Chargaff rule, is that the amount of A will be equal to thiamine, and amount of guanine will be equal to the pyrimidine. In together, it's also called as the amount of purines will be equal to the amount of pyrimidines. A plus G will be equal to T plus C. The immediate consequence of this rule is also that the ratio of AT, that is A plus adenine plus thymine divided by guanine plus cytosine, will give us a specific ratio. That ratio is called as dissymmetric ratio, and this dissymmetric ratio is utilized for the identification and characteristics of a species. Why? Because it usually varies from one species to another. One species will have it as one unit or one value, and another species will have it as a different value. So dissymmetric ratio can be utilized for discerning the species and to characterize a species in a particular hierarchical manner. So finally, we arrive at the concept of palindromes. Palindromes in DNA duplex or DNA sequences which are read in the same way in either direction. As you can see in the example, for example here, TENET, if you read it from here, then it will be read as tenet, and from backwards also it will be read in the same manner. These kinds of words are called as palindromes. In DNA also, if the sequence is like that, which is read in both directions in the same way, then that DNA sequence is called as palindrome. And it occurs because of the complementarity, when adenine will base pair with thymine and cytosine will base pair with guanine. And if there is a single stranded sequence of a DNA which has a complementary sequence of the same nature, which can be read in both strands in a similar way, then this sequence of DNA will be called as palindrome. For example, here the sequence ACCTAGGT, it will have a base pair of ACCTAGGC. So both will be able to be read in this similar manner in both directions. If you move from 5' to 3' in this strand, it will be ACCTAGGT. In the another complementary sequence also, it will be ACCTAGGT. So these are called as the palindromes. Of the palindromes is they are able to make a hairpin kind of a structure in sometimes. As you can see in the sequence here, you will see that these are the palindromic sequences GATC read in this direction or this direction GATC. They are able to melt and make hairpin like structures like this. So this GATC of one strand will bind to this and result in the form of a hairpin kind of a structure. These hairpin kind of structures are important in the regulation of the metabolism of the DNA. So this finishes the second video of the nucleic acid chemistry. Do visit my Amazon page for the books which you might like and be able to order. And do subscribe to my channel for further videos which I will upload in due time.